All right, in this video we are going to finish up our discussion of chapter 14, which was all about gene expression. And remember, in the first half we talked about information flowing through that central dogma from DNA to RNA to protein, and we talked about the process of transcription and RNA processing. And so today we're going to focus on the second half, which is translation. And then we'll talk just briefly at the end of the chapter about some modifications that might have to happen to polypeptides after translation in order for the proteins to actually be biologically functional. All right, so here is our by now familiar cartoon with the DNA being transcribed inside the nucleus, the pre-messenger RNA being processed, and then that full-fledged messenger RNA leaving the nucleus through a nuclear pore for translation at a ribosome. And let me go back real quick. If you look back at this cartoon, I think I pointed this out previously, you'll notice there are what are called T or transfer RNA molecules that are actually bringing in the amino acids to build this polypeptide. So the tRNA is really this adapter between the nucleotide language, which is in AGs, Ts, and Cs, or U's instead of T's in the case of RNA, adapter between that nucleotide language and the amino acid language of proteins. So this is really where that term translation is coming from, right? We're going from one language to the other. So we really have to have a special type of tRNA for each amino acid. And there's kind of two things that would have to happen in order to make sure that we're bringing in the correct amino acids to build the correct protein that's been specified for by that gene or that messenger RNA. So the first thing that would have to happen is the tRNA molecule has to actually read the codons in the messenger RNA. Because remember, it's those codons that are specifying what amino acid should come next, those three-letter words. If you remember the first half of the chapter. And then the tRNAs, they not only they have to read those codons, but they have to bring in the correct amino acid that should be specified by each codon. And we can see if we look at the structure of a tRNA molecule, here it is depicted in several different ways, but there's really these two key parts here that allow the tRNA to do both of those jobs. So the first, in terms of being able to read the codons, are these three nucleotides down here at the bottom of the tRNA, and these three nucleotides are called the anticodon. And so they would be three complementary bases. They would recognize the codon in the messenger RNA and actually physically bind to it, at least temporarily. And then you've got another three amino acids up here at the other end of the tRNA, which make up the amino acid attachment site. And so depending on which nucleotides are in this position, it may specify attachment of a different amino acid. So for sure, in any given tRNA, you would want to make sure you have those 6 nucleotides, quote unquote, correct. Okay, so just to give you a feeling for some of the steps in terms of following that genetic information through from DNA to RNA to protein, just to give you a feel for what that might look like. If I just focus your attention down here on the bottom of this slide, you can see here's, let's say, a portion of a gene, or maybe a very, very small gene, right, where you've got first codon, second codon, third codon, so on, right? That would be specified in the DNA. We transcribe that into a messenger RNA copy during transcription, and you can see the messenger RNA that we produced is complementary to the DNA, right? So instead of CAA, we get GUU, and remember we've got uracils instead of thymines that would normally pair with adenine. And then GTA becomes CAU, etc. So we've still got that code there, it's just the complement in the messenger RNA. And then those codons now in the messenger RNA are going to specify specific amino acids that should be brought in. So GUU, if we look at the genetic code, codes for valine. So we would want that amino acid first, and then histidine, leucine, so forth. And it's those tRNA's job to come in and actually read these codons in the messenger RNA and deliver those amino acids. So you might imagine, well, maybe you have to have a different tRNA for every single codon in the genetic code, but it turns out you don't actually have to have quite that many because of this phenomenon called wobble. And this is really the tRNAs taking advantage of the fact that we have this redundancy in the genetic code. So for example, down here, if we look at the amino acid alanine, there's 4 different codons that all code for that same amino acid. And the first 2 letters of each codon are exactly the same, right? GC, whatever, really, whatever the third letter is, it's going to be an alanine codon. So what the cell will do is it'll make a tRNA that has an anticodon that is complementary in that it will be CG, right, to pair with the GC. And then in that third position of the anticodon, there'll be a special sort of modified nucleotide that could bind to any of these bases. So we could really get away with just one tRNA that would recognize all 4 of these codons and then should bring in the correct amino acid. So how does the tRNA pick up the correct amino acid? And the short answer is there's actually an enzyme in the cell called aminoacyl tRNA synthase, which is a mouthful, but the job of that enzyme is to pair up amino acids and tRNAs. So here you see the enzyme in blue. It's got a big active site here where both the tRNA and the amino acid combine. And so usually the enzyme will bind the amino acid first. It'll bind a molecule of ATP because it requires some energy, input of energy for this reaction to happen. It'll remove some of the phosphates from ATP. So we go from a triphosphate down to a monophosphate. That sort of activates the amino acid, so it's ready to be added to the tRNA. Then what we call the uncharged tRNA, just meaning it doesn't have an amino acid attached to it yet, excuse me, comes in and binds to the enzyme. The enzyme attaches the amino acid and releases what is now the charged tRNA, which is ready to go off and deliver that particular amino acid. And so keep in mind, translation is happening in the cytoplasm on ribosomes, right? Ribosomes are the organelles that produce proteins. And I want you to think of ribosome as basically the workbench for building proteins. So ribosomes are not specific. You could make any protein on any ribosome, right? Just like on a workbench, you could make, any piece of furniture, let's say, right? So the ribosome is the workbench. And just to dive into the structure of ribosomes just a little bit, every ribosome is composed of 2 pieces, 2 subunits. There's the large piece, large subunit, and the small subunit. So kind of split this ribosome in half. And the way a ribosome would interact with the messenger RNA is those 2 halves will actually come apart, as you'll see, and they can kind of clamp down on the messenger RNA, as you see in the image in the lower right here. And then that messenger RNA can just slide through the ribosome, or the ribosome can slide along the messenger RNA. And both of these subunits, large and small, are made of a mixture of proteins that are really just there serving as a scaffold and molecules of what are called ribosomal RNA. And it's that rRNA, ribosomal RNA, that really gives the ribosome its enzymatic activity. And you'll also notice looking at this ribosome that there's kind of 3 slots or grooves here where tRNAs could be positioned. And they get these letter designations. So from left to right, it's EPA. And I always just remember Environmental Protection Agency. For some reason that's stuck in my head. You can come up with your own way to remember it. Um, but the A site, which is sort of the indoor on the ribosome, the A stands for amino acid because this is where your growing amino acid chain is going to spend most of its time attached to tRNAs in this A site. Um, P is polypeptide, so the chain is actually going to be physically growing in that P site. And then E is exit, so that's where the empty tRNAs are going to leave. So think of this as entry, polypeptide growth, exit. And here's what it would look like with a tRNA loaded into one of the sites here. And you can see the anticodon on the tRNA is complementary to the codon in the messenger RNA. And there's the amino acid that tRNA is brought along with it. So we've got to have that important match there. And it's really just random chance. So you can imagine there's going to be just a swarm of these tRNAs, all with different anticodons, just kind of fighting for position to get into these ribosomes. And if one comes in that doesn't have this kind of match, it would just immediately get bumped out of the way. And the next one would come in and give it a try. And so just like we talked about with the process of transcription, I wanted to just quickly go through translation and divide it into these 3 steps: initiation, elongation, termination. Initiation is obviously starting the whole process. A little tickle in my throat. And initiation looks like this. What will happen, remember the ribosome can come apart into its 2 pieces, is the small subunit of the ribosome will actually look for and bind to the start codon that's part of the messenger RNA. Remember, every gene starts with a start codon and ends with a stop codon. So it will bind to the messenger RNA, positioning that start codon kind of right in the middle site, the P site. Then the tRNA that recognizes the start codon will come in and bind. So anticodon binds to codon. And as soon as that happens, then the large subunit comes in and kind of clamps the whole thing down. So this is initiation, but I also wanted to point out that this is really the only time that the tRNA comes into the P site. So all subsequent tRNAs that come in are going to enter into the A site. And I also wanted to point out that even though they're not being shown in this cartoon, there are actually a bunch of other proteins that kind of help this process happen more efficiently, and they are called initiation factors. All right, then in elongation, where we're actually building the protein, it will look something like this. I'm going to actually start in the middle here. But here we've got a tRNA positioned in the P site, and it's got two amino acids attached. We're growing that polypeptide chain. So in will come a new amino acid into the A site. And then these 2 amino acids, the methionine and the proline, are going to get transferred from this tRNA in the P site over to this, become attached to the tyrosine to this tRNA that just entered the A site. So we move those 2 over, and then the whole thing shifts so that now this empty tRNA, right, it just lost its 2 amino acids, gets bumped into the E site and kicked out of the ribosome. This one that just came in and picked up the new amino acids is now positioned in the P site. And then we just do it all over again. So a new one comes into the A site, transfer these 3 amino acids over, shift everything over one position, kick the empty tRNA out, and we're ready to take in a new one. And so, really the catalytic part of this is that the ribosome is actually facilitating the formation of the peptide bond here between these amino acids. And I realize it's kind of a little bit hard to look at just in still pictures, so I'll try to find a decent animation to post on Canvas so you can see the things moving around. I think that will help. Um, but really, just like we talked about with initiation, and there being initiation factors to help that process, there are also other proteins that are not being shown here called elongation factors that would uh, help speed this along and make it more efficient. And so, yeah, as I mentioned, the enzymatic catalytic activity of the ribosome is that ability to form those peptide bonds, and that is really coming from the large subunit, and specifically the ribosomal RNA that makes up the large subunit of the ribosome. Researchers have found if you do something to basically destroy the ribosomal RNA in the large subunit, then the ribosome can't form peptide bonds anymore. Okay, so we would proceed through elongation, basically from the start codon all the way down the messenger RNA to the stop codon. And once that stop codon at the end of the gene slides into the A site, there is no tRNA to bind to stop codons, but there is this protein called the release factor that's just kind of hovering around, waiting for that stop codon. It sees it, it will bind, and that's like pushing the self-destruct or the eject button, and now everything falls apart. So the tRNA is kicked out, it lets go of the polypeptide chain that's been formed. That's then going to go off and hopefully fold up into the correct shape to make a functional protein. The ribosomal subunits come apart and let go of the RNA, and then we're kind of back where we started. And this ribosome, may clamp onto another messenger RNA and start translation again, or maybe even retranslate the same messenger RNA. So these things could all be reused. And a lot of times we don't see this in the introductory cartoons just because it gets a little complicated, but a lot of times you may actually have multiple ribosomes translating one messenger RNA at the same time. When this happens, it's called a polyribosome or a polysome. But it's sort of just like a stream of traffic. So you have a new ribosome just clamping on to the messenger RNA here at the start codon, and then you've got one a little bit ahead of it that's already got a short polypeptide chain, and then one ahead of it where it's grown a longer chain, etc., all the way down to one that's just finished. So it's just a little bit more efficient that way. We could crank out more protein in a shorter period of time. Okay, so hopefully you feel okay with that basic idea of how translation proceeds. And now I just want to talk for a few minutes about some things that may have to happen after translation in order to have a biologically active protein. And so one important part of this is actually getting the protein where it needs to go. So maybe the protein needs to be inside the nucleus, or maybe it just needs to stay in the cytoplasm. Maybe it needs to be sent to the Golgi and then to the cell surface to be exocytosed out of the cell. And so there's mechanisms that will help get these proteins where they need to go. Because if you make a protein but you don't get it where it needs to be, then what's the point, right? It's not going to be able to do its job. So let's focus on the pathway that would be we make this protein and we want to send it to the ER and then to the Golgi and then to the cell surface, sort of through that pathway that we talked about back in chapter 5, I think it was, yeah, chapter 5. And the way this works is it hinges on what's called a signal sequence, which is actually a part of the protein itself. So all translation actually starts out in the cytoplasm on free ribosomes that are floating around. But if a protein begins to be made on a cytoplasmic ribosome, and it possesses one of these signal sequences that will actually trigger that ribosome to move off or be escorted off to the surface of the rough ER, where it will dock, and then it'll finish synthesizing that protein actually into the ER. So remember, there's the rough ER and the smooth ER, and the rough ER is just covered with ribosomes that would be basically injecting the proteins that they make into the endoplasmic reticulum. And then from there, that protein, it might be modified in some way in the ER, packaged up in a vesicle and sent to the Golgi, where it might be modified further as it's moving through the Golgi, packaged up in a vesicle again and sent to the cell surface. Or, maybe there's a protein that needs to go into the nucleus or into a mitochondria or whatever. You could imagine lots of possibilities there. So essentially, this signal sequence, it says up here, is sort of sending the message to the cell, hey, or to the ribosome at least, hey, stop translation, go to the endoplasmic reticulum, and you can finish things up there. And they will literally dock, the ribosome will dock on proteins that are on the surface of the ER, these receptor proteins. And there's a little channel right through the middle of that receptor. It looks something like this. So here's the ER membrane. This would be inside the ER down at the bottom, and this is the cytoplasm up here in blue. And so here's the ribosome making this polypeptide, and you can see the little signal sequence, or that's basically like the first few amino acids of that protein. That signal sequence gets recognized by this, it's called the signal recognition particle. That makes sense. That's another protein in the cell. It binds to that and basically just drags the whole thing off to the surface of the ER where this docking process happens. And now that polypeptide chain is threaded through the pore in the dock. And once we resume translation, now this protein is going to get threaded through that pore and end up here inside the endoplasmic reticulum. And you'll notice there's a little enzyme here that comes in and actually clips off that signal sequence once it's done its job. It's usually not a important functional part of the final protein, so it typically gets removed. All right, so then we finish making that protein. It ends up in what's called the lumen of the ER, the inside of the ER. And there are some proteins that may need to stay in the ER. That's their job, is to hang out there. There's lots of enzymes that might need to be in the ER indefinitely. But typically speaking, proteins going through the ER, they might have sugars added to them. Something like that would be pretty common, become what are called glycoproteins, and then end up somewhere else, either at the plasma membrane or in a lysosome or something along those lines. There's a similar phenomenon. Instead of a signal sequence, there's what's called the nuclear localization signal that can direct proteins to the nucleus instead. But it's a very similar idea. It's not going to direct the whole ribosome to the nucleus, but it would typically send a fully translated protein there once translation was done. But this was a little experiment researchers did just to illustrate like how powerful these little signals are. In this case, a nuclear localization signal. So they were taking a protein that contained a known nuclear localization signal, the NLS, and they were basically injecting that protein into the cytoplasm of the cell. So there's tiny little microneedles you can use to do these kind of experiments. They don't look like this, but for simplicity's sake, we'll go along with that. So we inject that protein into the cytoplasm, and because it contains that nuclear localization signal, it all gets directed pretty quickly into the nucleus. In this case, it's got a purple tag on it, so we see it all in purple in the nucleus. We take that same protein and we engineer it so it no longer has those amino acids that make up the nuclear localization signal, and now none of it goes to the nucleus. We inject it and it just stays in the cytoplasm. And then just to kind of show proof of concept, you could take the NLS, that localization signal from this protein, and attach it to this other protein over here that normally doesn't have one and doesn't go to the nucleus, right? Normally this protein's found in the cytoplasm, but as soon as we attach that other signal, boom, it all goes into the nucleus. So, whether you're talking about a signal sequence, nuclear localization signal, uh, these things are very powerful, and they're going to make sure the proteins end up where they need to be. Okay, then, just a few other common modifications that might happen is maybe you make a protein, big long polypeptide like this, but in order for it to actually be biologically active, it has to be cleaved into, two or three parts. So that would be an example of what's called proteolysis. We take this and just cut it into these three parts, and now, one or all of these parts actually have some biological function. There's what's called glycosylation, where you take your polypeptide and add sugars to it. You can see them in red here. That's going to change the biochemistry of that protein a little bit and either add or remove some biological activity. And along similar lines, you could add or remove phosphate groups, so that might change its activity as well. And all three of these are really common things that would be happening in cells all the time, just to um, basically make sure that the proteins are fully active. So, all right, thanks for listening, and I will talk to you next time.